Okay, so let's continue our journey. It's lecture number 9, so we only 3 lectures left. So we continue to discuss stochasticity and randomness, and we use different examples where stochastic modeling is useful for understanding. So now it's a different story, and again, I encourage you ask me questions anytime, or we can talk after. So this is about transcriptional diversity. So transcription. We talked about transcription. So transcription, one of the most important fundamental biological processes. This is a process of reading DNA molecule and creating a corresponding RNA molecule. It's a part of transfer of genetic information. This is done by RNA polymerase. We already discussed these things for different problems. It's a complex chemical reaction, set of chemical reactions, which ensures that correct genes are replicated. It's probably one of the most studied phenomena, transcription, and still to a large degree not well understood. Already was showing this picture here. This is from a textbook. RNA polymerase should properly start reading DNA. So what is interesting about this thing that the RNA molecules that you produce actually do not live forever. The lifetime of our produced molecular RNA is actually relatively short. Different estimates gives this estimate between 1 and 100 minutes. So actually, closer estimates are closer to 1. So what is the reason for this? It is needed to remove the defective RNA species as well as to save the material energetic cost. So that's one of the reasons. So you cannot always keep RNA, you have to remove them. And that's the reason. And different ways of removing degradation are shown over here. We don't need to look at the details. The only thing we have to remember that there are different ways of removing RNA. But then comes a very question. Okay, so remember, what is the main biological dogma? You have DNA, you're producing RNA, and from RNA you're creating proteins, and proteins build everything up, right? So if you think this way, and if you want to be naively thinking biological system should be efficient, you would think that RNA should be always produced. Okay? So the question is, RNA production continues? And surprising answer is no. Despite the fact that you always need RNA, they're not always produced. Okay, so this is analogy here is, suppose you need to make apple pie. Okay, you need apples, right? But for some reason, you occasionally just throw away all your apples to the trash can. That's how it looks like. The fact that there is a production of RNA in biological system is not constant. How do we know this? That's because people experimentally observe something which is known as transcription burst. So this picture shows experiments where people were able to measure at single cell level number of produced RNA. And you can see here number of RNA. 1, then there is a jump, 4 produced, and there is nothing, and then disappears. 2 produced. In other words, there is a discontinuous transition. So the RNA is not always constantly produced, it's removed frequently due to degradation or something. And this is a phenomenon known as the transcription burst. So there is more, another experiments which show the same thing. So this is experiments on genetically identical mammalian cells. So this experiment, they took identical mammalian cells, absolutely identical mammalian cells, and each of them were able to calculate RNA production, and it's not the same. There is a distribution of produced RNA molecules, and you can estimate the mean as well, something called Fana factor. We will be talking in a moment. So there's a significant variation in the number of produced RNA molecules. And this is due to this transcriptional bursting. It's essentially stochastic effect. Another experiments in the molecular experiments where you, when the RNA is produced, there is a fluorescent signal. You can see this is RNA produced, and then there is nothing, no production, RNA produced, and there is nothing. And you can see that the time scales over there. So again, so there is a RNA molecules production is not continuous, despite the kind of naive expectation that it should be. It's one of the most important molecules. Okay? So, what are the problems? The problems, there are many problems. The problems, if you look at this graph, you would actually even understand the problem. So, the time RNA produced is of order of 100 seconds, and then there are seconds, 1,000 seconds, nothing. So the time of production is even smaller than time when RNA is not produced at all. Okay, then there is a question: what is it? What is the source of such stochastic behavior, and what is the purpose? Why nature does it? Okay, because we talk about many things, and nature seems to be optimizing things for something, right? Not all, we don't know exactly for what, but this seems to be counterintuitive. This is one of the most important molecules, and you're not producing it frequently. You're stochastically changing production or non-production. Okay. So in trying to understand this phenomenon, people introduce models, and that have the confusion. So people call this multi-state kinetic models, but I put it in a quote, and let me try to explain in a moment. Okay, so let's start with the simplest model. The simplest so-called one-state kinetic model of RNA production. It's not actually one state, it's a one pathway or one channel of RNA production. Imagine that these numbers tell you how many RNA molecules are produced. RNA molecules produced with a constant speed because we're just reading the gene. Gene is always here. But then they degrade. The beta is degradation rate. And rate constant. And there is two produce and two rate overall and so on. So there is a degradation rate proportional to the number of RNA molecules present. So this is, although this is called the one state, it's actually one pathway. This model describes this continuous production. There will be always production. So this explanation, this one state is actually not a one state, but it's more like a one channel or one set of biochemical parameters describing this. This model can be easily solved, and you can calculate, for example, this is stationary probability to find the system in state, and it's a Poissonian process. I'm pretty sure you took the simplest class, simple Poissonian process. You can easily calculate average number, what will be the average number of RNA in this process, or this measure of fun factor, which is a dimensionless measure of fluctuation. And this parameter X is the rate, essentially equilibrium, effective equilibrium production constant, is the rate of production over rate of degradation. Yes, so important result here that this model always predicts this fun factor to be 1, right? So then you can think about how good is this model for experimental data, and it's bad. So you see, this is the fun factor measured experimentally, and it's essentially in most of the cases it's not equal to 1 at all. So which is indication that this one-state model, one channel of production does not work. So you need to have a multi-state kinetic model. You need to have multi-state kinetic model. So that's a multi-state kinetic model. So it's multiple channels of RNA production and degradation. And of course, this is stochastic model, but multi-layer, multi-channel stochastic model that I'm arguing you have to use to describe transcription growth. I hope you understand, right? So you can have one way of producing RNA, the system can switch to another way of producing RNA, to many other ways, and there's always, in this direction, there's always degradation, in this direction, there's always production. This is the general picture. So essentially, this you can solve this analytically or simulations or quite well numerically. You can actually try to understand the system quite well. And what you try to do something like this, this is a distribution. You can calculate distribution from simulation to analytically. And what is interesting is that, let's say this is for the case of 3-state model. 3-state means there are 3 channels. And depending on conditions, you might have 3 peaks, 2 peaks, or 1 peak. Okay? So despite of the fact that originally there are 3 different ways of producing RNA, in the distribution you will not always have. In the distribution, you will not always have peaks. So you understand, each peak corresponds to RNA copy number, right? And each peak in principle corresponds to different channels of production, but it's not always 3. That at the beginning it seems to be surprising. And this is a kind of a dynamic phase diagram. shows here, only here there are 3, here will be 2, and here will be 1.
Professor, so here I wanted to ask about the states or the channels as you're calling them.
Right.
Is it one system which switches between these channels, or are these 3 channels operating in parallel?
No, the system switches between.
Okay.
So it's so if you want to do more microscopic picture, so there is gene which producing RNA, but maybe there is some protein sits to gene and blocks it, and there is no production of RNA or weak production, very different channels of production. So it's not simultaneous. Okay. So, but if you think a little bit, it's not so difficult to explain these different peaks of observation. That's because if transition rate between different channels are very fast, then you can effectively go in effective equilibrium. If transition between, let's say, state 1 and state 2 are very fast, then you cannot distinguish this to state 1 and 2, right? That will be, you cannot have different peaks. And then if transition between all of them very fast, the whole system behaves as effectively single state. And that's the explanation for this, for all these observations. There could be 3 peaks, 2 peaks, or 1 peaks. And then generally it could be m peaks, m-1 peaks, and n-. Okay, good. So this stochastic model, so it's a multi-channel stochastic model of transcription work. You can do a lot of things here. And again, one of the questions you can answer is the following one: how many biochemical states you need? How many channels you need to describe transcription work? In other words, we can have model with many possible channels, but what is the best description? So I already argued one channel is not good. Continuous description is not good. But what is it? 2, 3, 4. This stochastic model can argue that that you should have relations between number of these channels and funnel factor and average number of produced RNA. There are experimental data, and we put experimental data for. So this is for E. coli bacteria. They measured this for many different genes, for hundreds of genes, I think, here. And this is estimate of M from this formula. What you can see, the majority of the cases is, or this function of average N or average F, for the majority of cases, 2 is a very good description. For the majority of cases, the so-called 2-state kinetic or 2-state stochastic model provides quite an adequate description of transcriptional version. So you understand, each of the point, different gene. It's a different gene, different RNA. So that's what description. So this is a very nice picture, but the problem you have to understand that this stochastic model, despite the fact that already non-trivial, is a huge simplification already, because there could be some other biological phenomena. It's a genetic regulation, so-called feedback regulation, and this might affect it. But as the simplest possible approach, this seems to be reasonable and explains data, and it's enough to have 2-state production. This 2-state production is one state when gene is on, you're always producing RNA, and gene off, you're only degrading RNA. Seems to be very okay. Good. So this stochastic model which can describe this observation, but the question is why sometimes you have on, sometimes you have a why sometimes you're producing RNA, sometimes you go off. That becomes unclear. But extension of this stochastic approach can provide you something, can give you some explanation. So more specifically, the question is what ends the transcriptional birth, right? You're producing. Suddenly, why you stop producing? Why your RNA polymerase reading? What makes system to stop transcription? And that's what we can do is we go back to single molecule experiments, and single molecule experiments argue the importance of topological and mechanical properties of DNA during transcription. So that stimulates so-called the development of mechanochemical stochastic models. So let me try to explain you what do I mean by this. When you probably know when transcription happens, this RNA polymerase doesn't move linearly. It's actually does rotational motion. So it does rotational motion. It's effectively supercoiling DNA, and if 2 ends of DNA are frequently somehow attached, you are creating a problem. Okay? I don't have a model, but imagine that if you are doing something, right, you're screwing, you try to do it yourself with the roll, right? At some point it will be difficult. You understand, right? I don't want to throw my clothes. So this is a point of mechanics, right? You can do this process, but every time you rotate something, it becomes harder and harder. So when you're building up mechanical stress, maybe you still have stationary phones at home. You remember that because the wire, maybe not anymore, advanced, right? Then they have this already, and you can really sometimes supercoiling. There was a problem to uncoil. Anyway, so this is the problem. This supercoiling can resist mechanically resist transcription, and you can obviously couple chemistry. Chemistry is a transcription and mechanics. So there are enzymes known as the topoisomerases, which actually can correct it. Biological molecules are polymers. Okay, and polymers are prone to do these things. There's a huge area of knocking. I think somebody was working even on this, if I'm not mistaken, student. So there's a lot of topological issues come out in biological system, and nature is aware of this, so it uses a lot of this enzymes which correct the problem. For example, there's also supercoiling. You can do this, and you can uncoil it. It's a big deal here. Okay, so the same with the transcription. There is a so here there are topoisomerases that can uncoil, that can help to remove the stress. And that's a stimulus for us to develop a model, which is I call mechanochemical stochastic model. So it's mechanochemical, chemical because it's making transcriptions a chemical reaction. It's a mechanical because, as I said, every time you're doing supercoiling. So mechanics opposes chemistry in this case, and you can do this model. So the model is, in extension to what I told you before, the model is relatively simple to describe. So your system could be in a state on, you're doing transcription, but simultaneously this enzyme present, GIAN. GIAN is enzyme topoisomerase, which immediately corrects the problem. Okay? So there is one possible channel of production, you're doing this and supercoiling immediately removed. But GIAN can also dissociate, it's not always present. And in this case, you will have this reduced rate. You will try to do transcription that will be harder and harder and harder. Okay, so that's this state. And then that's what is going on here. When you do supercoiling, it will be harder and harder and harder. And schematically, you can do it as this model this way. So this is the production when state is on, there's no supercoiling, and all other channels are with different degrees of supercoiling. So J is the degree of supercoiling. And there's the rate of production, and you can take into account the supercoiling affects the rate of production. So that's exactly what I described. Here I have essentially fast transcription rate. There is no mechanical problems here, but here I have this doing transcription against supercoiling, which is not good, and the stronger supercoiling, the weaker the rate. And that shows this. So you see that more supercoiling, the larger this exponent with the parameter y, which the stronger is the opposition to production. Yeah, so that's what I described here. This is effectively the rate of RNA production from state J, and J is the degree of supercoiling, and you can associate this with effective energy, how much energy you add due to supercoiling. And that quantifies how mechanical forces are resisting chemical reactions. And you can solve this model analytically again, surprisingly. You can do a lot of things here. And you can calculate, for example, this is the probability of having different number of RNA molecules, so different set of this supercoiling coupling. And you can see there is no supercoiling coupling. You have this orange things here. But if there is some supercoiling coupling, there will have some distribution. And the stronger there will be, the less you have RNA, and more in state 0, there is no RNA production. But yes, and you can also also calculate in this approach distribution probability of transcription as a function of degree of supercoiling. Just have to change the parameter. And this parameter is the rate of G-raised binding. Okay, so let me remind you again. So what can save this problem? G-raised binding. If G-raised binds, you immediately go into this good state. Okay, so K1 is the rate of G-raised binding. So if you increase the rate of G-raised binding, that's right, you immediately can solve the problem. Save the system. Okay, good. Yeah, so that's what also we showed here using the stochastic model, that you can change the degree of supercoiling. So the parameter y is, you can think about it as a spring force, right? The larger y is the strongest spring which opposes the motion, if you wish, which is the dimensional parameter. Yeah, so increasing this mechanical coupling, this parameter y increases the effect of slowing down due to supercoiling. And you can really describe system quite well, but the most important things with this approach that you can explain why there is a stop of transcription happening. You understand, right? So you can go to mathematics. So the biggest problem was that transcription is going on and suddenly it stops. This picture provides a possible reason. That's because you supercoil too much and you cannot go, you cannot squeeze spring too much, and like the system stops doing this. That's the value of this model, of this approach, stochastic approach. So you can do, so you can compare your theory with in vitro measurements of transcription growth in this E7 and E. coli bacteria, and you can estimate this energetic also supercoiling for each term, and energy turned out to be relatively weak. So you can think about DNA as a relatively weak spring, but still, if you do a lot of supercoiling, eventually spring becomes strong, right? So that's the description of experimental data. And yeah, so we can do this thing. So this analysis suggests that supercoiling is relatively modest, right? And if you think a little bit, this is a very good point from biological point of view, because if it would be too strong, you would just stop transcription immediately. That's not good. You need transcription. If it would be too weak, that's not okay too, because you need a control. This is everything about the control or regulation in the biological system. Everything about the regulation. Okay, so essentially, I give you a relatively simple stochastic story of transcriptional bursting, right? And we show that you can really get some important insight in the stochastic approach. We also explained the possible picture of transcriptional bursting. We presented with experimental data, everything. But let me ask you the question. So I already posted the question, but didn't answer. I want to hear you. So one of the questions was why nature goes into this complexity, does transcriptional bursting? Why nature does not make sure that the production of RNA continues, maybe slower? Why do you think it does these things? Why? What do you think? So let's repeat again. So one possible scenario would be that nature could make sure that RNA production is constant, more or less constant, all the time, right? But nature doesn't do it. It goes into this, in my opinion, quite a complex situation when it's on, off, right? If you're in a room and you want to save the light, right? You just buy a more energy efficient lamp, right? You don't do like on, off, right? Why nature does it? What do you think? What is the most, I already mentioned, what is the most important things in biology, in this system? The ability to regulate, that you can regulate, that you can control things, right? To control things, you have to be able to quickly change something, okay? So on, off allows you this to quickly change, okay? Government needs to control you. How? So they can do something, give you more money and give you more taxes, something like this. It cannot be continued, otherwise it's not possible to govern. Biological systems are frequently optimized so that they are regulated the most efficiently. Regulated means that they also respond to external signals quickly and efficiently. It has to be robust. Constant production of RNA would not be robust. Make sense? Surprising things here. So that's what probably is the case. Yes, again, it's another story where stochastic picture works reasonably well, right? reasonably well. You can describe things, you can understand many things here. This story, in contrast to what I told you earlier in the morning, shows that this stochasticity is needed. It's needed for better regulation, right? So the story before was you can couple stochasticity to produce something very precise. No, this is not the case here. You do stochasticity here to better regulate things, to control things better. Okay? Okay, there is a lot of biology involved. I try to eliminate all possible complexity. There is this very interesting systems which, okay. Yeah, so this is the end. If you have any questions, please feel free to ask. Yes, please.
So how is this works validated? Like it is validated with respect to some mice model or something like that?
There is experimental data. You can feed experimental data. Okay, you can feed experimental data. You can explain these things here. For example, yeah. So we got it. Okay, didn't plot it, but you can get it in the paper. We got experimental data. We can describe reasonably well. Here, the most important things was. So there is observation on, off, but the question was not clear how system goes to off. Why? And that's a possible reason, mechanical reason. You're just hitting spring too much and system just snaps out.
So this experimental data is taken from?
There is single molecule experimental data which is coming over there. Yeah, so from this experimental data. There's a single molecule experimental data when they looked at the transcription on specific things, or they had also this supercoding thing.
So there are some DNA bound structures also like histones and so, which
okay. In a typical physics modeling way, I throw away all this complexity. I think it's not important. But you're right, it's adding additional complexity. Which adding additional complexity. Okay.
Do we have something like G-rays that can?
Yeah, so G-rays is one of those things which is a topoisomer which corrects topological things. One of those, it's not over there, but one of those things here. So you see, if you create structure like this, the G-rays, for example, one of those related topoisomer allows to. So it does something like this. It simultaneously takes two pieces of DNA, cuts something, then exchanges and cuts it and connects it back. Okay. My picture here was, you see what it is? So suppose there is a kind of a knot here, right? You don't want to, what you do, you cut it suddenly, and then you really like by hand change it. Everybody has problem with knots, right? Especially when we're kids. So this is a knotting problem. It's a knotting thing. So it's done quite nicely. And the result, you get something without, topological problem. Topological problems are completely different.
You had a picture of the stochastic model. A little ahead, you had those diagonal arrows. I was wondering what they represent.
Okay. Yeah, so this mechanical model, right?
Yeah.
So this mechanical stochastic model. So this is again number corresponds to RNA molecules in your system. 1, 2, or so, right? You can produce more RNA with rate alpha here in state of alpha, right? Or you can degrade it. So this is exactly the same as I had before. But then, and this is there is no any problem here because G-rays immediately corrects the supercode. But you can be also in a state which is identical to this one, but there is no G-ray, right? If there's no G-rays, you need one circle, and you are actually you created from here to here, you created one RNA molecule, but you simultaneously increase the stress. Supercoding, right? You created one molecule, you increase supercoding. You created from here 2 molecules, you increase supercoding. That's why it's a diagonal transition.
So the higher J represents more supercoiling.
Yeah, more supercoiling, more stress in the system.
So from J equals 0, you can have a transcription event, but the cost will be that you have more.
That's right. You have it, but the cost increases more and more and more. That's why I call it mechanochemical. So you couple chemistry with mechanics.
The second thing is probably not so much related to this. Is there anything about translation being bursty, or is that? I know that's a different story.
It's excellent question. There is translation also. Okay, translation is the process of synthesis of DNA. There is also fluctuations, but it's still unclear are these fluctuations due to fluctuations here, or there is additional sources. Because there is a difference between transcription and translation. In transcription, you need to have gene in active state to make RNA, right? And gene can be on or off. In translation, as soon as you have even at least one RNA, you can do synthesis, right? You don't need, you can have 5 or you can do 4. You will always produce protein with a different speed. So there is a qualitative difference between translation and transcription. So
One is any, you want to say something?
yeah. So this is a story where transcription is really, randomness is really needed. Okay. So I was trying to, all many stories that I told you before, transcription or stochasticity was used to kind of reduce, right, to do it precise. Here, completely opposite. Randomness is needed here. Okay, so that shows a multifaceted phase of stochasticity.
Yes, one more thing. Does the term bursting come in because it's off for extended periods of time?
Yeah. Now, right, let's go back to experiments of those. Yeah, so the single cell experiment. Yeah, okay, maybe this one is good. So you see there's no RNA production, and suddenly you're producing RNA, and then, oh, no production, no production, producing RNA, no production. Or you can go to another, this one, for example, this single cell measurements, how many RNA in each cell? 1, then suddenly 4 added, and there is nothing added that even actually decreased. Because when there are 2 added, you see there is this, it's not going smoothly, it's like jumping, which is clearly stochastic phenomenon. We don't know why it's jumping at these specific periods, but that's what it is. So sometimes you need to be stochastic, right? The governing party should not have strict rules, it should be flexible, okay? Depending on the situation, you can change your point of view so you can win the next election, something like this. Regulation is important thing. Anymore? Yeah, supercoiling stress, I use it as a synonym. It's the more supercoiled, the harder it moves. Because the way RNA polymerase works, it's going like this, it's rotating. So every time, and it's harder if it's harder to read DNA, then its transcription rate will go down.
No one has any questions. Let's thank the speaker.
The addition of the thing goes slightly, but the decrease, there is a sudden drop. So why so? In the addition part, like there is a +4, the addition is a slow. But in the decrease part, there is a sudden drop.
That's actually a reflection of the fact that synthesis is probably slow rate, but degradation is fast.
So there can be a slope also in the negative.
Yeah. But typically degradation is fast. It's typically fast because there are so many ways to degrade the RNA. See, there are so many channels to degrade the RNA. All of the different things. Okay. So important things here. Sometimes it's better to system to be random, and that's better for control. By the way, this idea sometimes used in this mobile network, right? It's not always necessary to be very, very stable. So fluctuation sometimes useful because then we can change something quickly. Okay? It's a different story.
So doesn't it sound a little bit strange that being random?
It is. That's the reason I'm telling you this story. So here is stochasticity is important
But in
to keep things regulated well.
Yeah.
So to eventually have it almost deterministic outcome, but in non-intermediate states you need to be stochastic.
Thank you.
Okay. Thank you. Okay, sure. Okay, sure.